It's the disease of our time. So complicated, treatment can be a guessing game based on percentages.
Most patients are still going through chemotherapy and even radiotherapy, and sometimes they do not respond to these treatments.
The answers that we are giving to them are not the ones that they would like to hear.
Now, one scientist and her research team are seeking to remove the uncertainty.
This type of research will be enormously important because we can individualize the treatment for the patient facing us on the clinic.
Cancer is a word we all fear. It's often described not as one disease, but many, because every tumour has a different set of mutations, which makes it harder for doctors to determine what the best treatment will be. I'm Emma Keeling in Lisbon, Portugal, where a more personalised medicine is being enabled by a tiny helper. Professor Rita Fior's mother was diagnosed with lung cancer in 2009.
And I was discussing with a friend oncologist what was happening and which treatments my mom was getting. And then she told me that in another hospital they don't give that drug anymore, they give another. And this was really a shock to me at this point, because I was not expecting that. I was expecting that all patients with that sort of tumour, that stage of tumour, would be treated the same.
Yeah. So it felt like it was a game of chance with your mum.
Exactly.
So is there no test to say whether you're going to react well to chemotherapy or radiotherapy or any other medicine?
No. Well, there are some tests, but not for very specific therapies, but not for the general chemotherapy and radiotherapy that most patients go through. So there was no test at the time, and that's why we started thinking that maybe the zebrafish could help us.
Yes, she did say zebrafish. Rita was already using them in her work as a developmental biologist. Zebrafish and humans share 70% of the same genes and 84% of disease genes, and have been used in almost every area of medical research. Her idea was to use them as test animals or avatars by implanting each patient's cancer cells into many zebrafish, then treat each group with a different therapy to see which responded the best. Oh wow, look at all the fish.
So this is our tanks.
So this is your, these are your babies?
Yes, well, adults. So these are the breeders that we cross females and males to have embryos, and then they grow to little larvae, and these are the ones that we inject in the lab upstairs.
So are you the first to work with zebrafish as avatars for cancer?
No, so when I started, I had this idea, but then I went to the literature, and there were people that were pioneers and really started. It wasn't fully optimized. The big sharks in the field were not really believing much in the model, but I wanted them to review my paper.
Oh, so they thought
to see.
They were dismissive before, and so you turned up and said, hang on, read this.
Yes.
So you decided to go to the ones who really did not believe in this model.
Yes,
You
I
wanted them to
review my paper.
Feisty, I like it.
Yes.
Good, good. And they've now changed their mind about the zebrafish.
Yeah, I think so.
Yeah. I mean, you've already done some tests, haven't you, using human cells? Has it been positive?
Yes, really promising. So far, so we started with colorectal cancer, where we have tested more patients, and we have around 81% of predictability, meaning that we can predict or even guess, well, what is going to be the response in the patient, and 100% in breast cancer.
Wow. To understand how hard cancer is to treat, you need to understand how it operates. Cancer is caused by changes in our genes. These can be inherited or happen as a result of errors that occur as cells divide, or from damage to DNA by environmental factors such as smoking or the sun. Cancer is complex because the cells have more mutations in their DNA than normal cells, and as the cancer grows, more changes will occur. Even inside the same tumour, different cells may have different genetic characteristics. There are around 24,000 genes in the human body. If two people have only one gene different in the same type of cancer, the same treatment may not work for both. For oncologists or cancer specialists like Dr. Joanna Ribeiro, Rita's research will hopefully provide more certainty, helping to give the most effective treatment the first time, sparing unnecessary side effects. So you must be so excited about Rita's research because it means you can actually personalise treatment.
Yes, it's very exciting because we as clinicians, we face this problem when we are talking with the patient. So what therapy do you have to offer me? And then it comes the question, how do you know it is going to work? What are my chances? And our answers are completely based on big studies looking at heterogeneous groups of peoples that give us a probability for a group. But they don't tell us anything about that person in particular. So it's like when you have an infection, you go and you do a blood or a urine sample, and then you go to see which antibiotic the bacteria is sensitive to. And we don't have anything like that in cancer. I think if it works, it will be a major advance in the way in which we talk to patients.
As far as lifesavers go, the minute zebrafish larvae don't look like much. But like everything, it's what's inside that counts. A tropical freshwater fish, the zebrafish larvae work well because they are transparent. Separated from humans by 450 million years of evolution, we not only share 70% of our genes, we also share an immune system, many organs, and have a similar sleep-wake cycle. This is why these tiny wonders are ideal for genetic studies, which have led to advances in cancer, heart disease, and regeneration studies.
So we're going to see the injection. So how do we inject human cells into the zebrafish, human tumor cells into our little larvae? And it has to be done with a microinjector, very thin capillary, and under the stereoscope because the larvae are really, really small. First, we inject the tumor cells. So then you'll have the fish with the tumors from a patient. But you'll have like 200 fish or 100 fish, imagine.
I can hear a tack going on here.
That's injection.
Okay, can you talk us through what's...
Yes, so here is Kel. She's injecting breast cancer, right?
Yeah,
breast cancer cells. And so this is the needle.
Oh, wow.
And then there it is. So these are the cells injected in the fish.
I just want to say that what Raquel is doing here, this is a pinprick of a fish, and she's somehow getting under the skin and injecting thousands of microscopic cells. I mean, this is she like your sniper of injectors?
Oh yes,
she's
for
like
sure.
a sniper injector. There's
sure.
genius going on here. It's incredible.
For sure.
So how many different fish do you have per patient, and how many fish per different type of treatment?
If we have a big sample, if it comes from a surgery, we can have, in the end of the experiment, around 30 fish to analyze, or 40 to analyze. If it comes from a microbiopsy, then we have less fish.
It doesn't seem like many. I mean, you must be a bit worried about...
It doesn't, but if you think about mice experiment in the mice, it's like 3 or 4.
The day after cell injection, the larvae are checked.
We can see if we have the tumor cells inside the fish. Then we start distributing the fish in different conditions. Control with no drug at all, so normal as a patient would not have been treated, right? Or with drug A, drug B, drug C, or we take them to radiotherapy, for instance.
As we'll see in part 2, the group that responds the best indicates the best treatment for that patient. Professor Rita Fior and her team are creating a personalised test for different cancer therapies using zebrafish larvae as test subjects or avatars. Cancer patients will often come here for radiotherapy, and where the people go, the zebrafish larvae follow. Would you say this is the smallest patient you've ever had to radiate?
No doubt of that. The fact that we can select patients depending on how they will respond to different strategies means that we are actually avoiding toxicity to those that they will not respond to that treatment. And that's very exciting. Actually, science these last years have been investing a lot of time and resources on finding biomarkers to predict response to the patients. so far with not that very good results. So we are still on the same starting point. All patients will be treated the same way. And this is giving a little bit of hope and light to be able to tell a patient in a very short period of time, okay, we will avoid this because this will not work on you. We will go to another strategy of treatment because it's likelihood you get better response.
As any patient will tell you, waiting to see if the cancer responds to treatment is agonising, which is another reason why this research is so exciting.
We can see if the cells are being affected by treatment in 4 days and then quantify how many cells are actually dying with this drug.
And then you know whether
which one is the best.
Whether A, B, C or D. How long does it take to analyse the data?
So overall our goal is to, in 2 weeks, have the result. 2 weeks.
That's so fast. How long does it take to get that kind of data from a mouse?
Oh, months.
The difficulty of the treatment decisions cancer doctors make is more obvious when you put the cells under a microscope.
And here in these pictures you can see a normal tumour with no drug, so it's control, we call it control.
So nothing is dying?
Nothing. So you have a few cells dying in green, very few because cells die just
there's no
this
drug.
is the basic. But if you put drug A or drug B, you can see much more green here, but much more green here with this drug. Okay, so this has much more effect than this one. And if we look at another tumor that has different mutations, there's no effect of the drug. Although it's exactly the same drug, almost has no effect.
Cancer cells can seek out a blood supply to grow, called angiogenesis, which is a key event when tumour cells spread to different sites in the body. It's difficult to know when this starts, but in the tiny larvae, researchers can see when a single cancer cell moves through the body. It's so complicated, cancer, isn't it?
Yes, that's why it's so hard to treat, because you can have 1, 2, 3, 4 major mutations, but then these mutations interact between each other and can have a different output if you just take one out, and it becomes really complex.
The results are promising so far, but the team has only analysed 21 colorectal cancer patients and 6 with breast cancer. More are needed to be confident in the avatar model. Fortunately, there is no shortage of zebrafish. So are they breeding all the time?
So the breeding, each tank breeds once a week.
Okay.
More or less, in average, once a week.
Okay. And
so
how many eggs are we looking at there? What, hundreds, thousands?
Thousands of eggs that we have every week. Yeah,
wow.
To inject.
That's great.
That's a big advantage of the zebrafish, is also the amount of embryos that you can get from a breeding pair.
So, I mean, they are just the winners when it comes to avatars, aren't they? I mean, mice, out of the way. Zebrafish are the way forward.
Well, I don't know if you should say that.
Okay, don't say that. Let's pretend I didn't say that.
No, it's okay, it's okay. We can say that because the mouse are great, because they're a mammalian, and so it's the gold standard, and we've learned a lot from the mice. But for this purpose, which is the avatar and to help treatment in a short time window, zebrafish, and they're much better.
The zebrafish facility is located a short walk from Rita's lab at the Champalimaud Centre for the Unknown, a multidisciplinary institute. Being on site with oncologists and surgeons, as well as fish, has allowed a clinical trial collaboration not usually found. So some of your patients have been taking part in the trial. What's their reaction?
Well, I think the reaction is they are really, really interested in the results, and most of them, they want to learn what is the result of their tumor in the zebrafish. Of course, they understand that they cannot refuse, so far, or even accept different treatments just because of these results, because they are still early phase results. So it's really, really a very interesting connection between the patients, the surgical or even the multidisciplinary team, and the fundamental researchers who are doing this. It's really good.
If we are able to personalize therapy, we can do smaller trials just looking at the drug that seems the right one for those group of patients, and we will have a response much faster, and we will able to get these new drugs from the research arena to the clinical practice faster.
Your mum is no longer with us. Does that sort of spur you on, thinking of the frustration that you went through when she was first diagnosed?
Yes, that's if you think thinking about my mother, or thinking about the technology that we are surrounded every day, scientifically, we can go to the moon. People are thinking about Mars. Every day we can sequence genomes every day, but then if you have a cancer, you don't really have a test to help those patients there. But something has to be done to solve this problem, and we're trying, and that's really our drive.
Think your mum would be proud?
I hope so. I think so.
It's hoped that with most of the testing being done on the zebrafish, doctors will be able to give patients the therapies more likely to work.